everyone, welcome back to Alkaline Hydroxide 784, and today in the chemistry series we are going back into organic chemistry, which is isomerism. So that's the second part in organic chemistry 11.2, and actually I just realized a bunch of the learning outcomes are literally just statements that you need to know. Okay, so let's get right into them. The first one is describing constitutional or structural isomerism. So structural isomerism is like the simplest thing, or at least the first thing that you probably would know about, which kind of happens when you have, the like earlier we saw the difference between just the molecular formula versus the structural formula, and from there we can kind of tell that the way that the atoms are arranged makes the molecule different, but at the same time it has the same stuff in a way. So this is generally used, like all of these are used for organic chemistry generally, although it could apply to other stuff as well. So in organic chemistry, the most common kind of constitutional or structural isomerism is basically just the functional group being on a different part of the molecule. Instead of maybe at one, you have it at the second carbon or something, instead of the first, instead of the second, or maybe the third carbon. Okay, it depends on how long the carbon is, like the carbon chain, because technically if you only have that one functional group and your carbon chain is, well, an odd number or something, or even like even number, odd number, whatever it is, depending on its symmetry. If, like, imagine you number it 1 to 5, putting it on carbon 1 is the same as putting it on carbon 5, because it's usually you're not looking at double bond here, it's just completely single bond, and then you just change the location of the functional group. So yeah, you must be aware of what exactly counts as a separate isomer. So as for constitutional isomerism, it's usually just either you introduce extra branching, like for example, instead of having a butane, you make it a propane with a branch in the middle, or you can have a different placement of the same functional group. So that's the kind of stuff that you encounter in structural isomerism. It could also just be like, maybe you completely reorganize the stuff, but it still has the exact same formula. So yeah. So the next one is starting to get a little bit more interesting. So you have cis-trans isomerism in alkenes, because you need a double bond to have a cis-trans isomerism to begin with. So that's the first thing you need to know. You have to see whether there's a double bond, and the double bond will have like 2 carbons usually, and the 2 carbons will then have a bunch of stuff attached to them, right? So they have 2 different things attached to them apart from each other, and those 2 things you must have you cannot have like how do I put it? Yeah, you cannot have like 2 of the same thing attached to 1 carbon. So if, let's say, you have each carbon has a hydrogen and a OH attached to them, so maybe CH and OH, and then the other one also has a H and a OH, then you can get cis-trans isomerism in it. But if, let's say, one of them has 2 H's and the other one has a H and a OH, since the first one has 2 H's, it doesn't have any isomerism anymore, because it's the same thing either way. Yeah. So to actually get the isomerism, you need 2 different things on functional groups or whatever it is on the carbons. So how it works is, for the cis isomer, you have both of the same thing on one side. So you imagine, like, if we go with our current example, we have both of the OH on the top, like when you draw it. Like when you draw it out, you draw it both on the top, and then the H will be both on the bottom. And trans would be, for one of them, the OH is on top, H is on bottom, then the other one is the H is on top, OH is on bottom, so it's like a diagonal kind of thing. So that's how cis-trans isomerism is drawn on a piece of paper. But if you want to do the stereochemical way of drawing it out, you can basically use it. You can just use your perspective. Based on your perspective, you can tell which one is sticking out, which one is not sticking out. So the other place where you can find cis trans isomerism is in a ring. So because in a ring you can't really rotate it around and make it the same thing again. So it's actually kind of like Cairo. It's kind of like the Cairo stuff as well, but in a way you can do cis trans isomerism on a ring as well. And you can use a stereochemistry to make it look more obvious, but where each of the the functional groups or whatever it is at in the sense. Yeah, so I'll hopefully have drawn an example here. Okay, so the next one is okay, before we move on, the reason why this isomerism exists is because in a double bond we have that pi bond, right? So the pi bond is from a side-on overlap of the p orbitals, and therefore you can't really rotate it much. Like you imagine, the p orbitals have like the top part and the bottom part, and both of those connections there equals to one pi bond. So if you rotate it in any way, the thing is gonna break. It's not as strong as the sigma bond. Therefore, it doesn't allow you to rotate it in a sense. It doesn't make sense to rotate it. Otherwise, you can't really maintain the pi bond. So yeah, that's why it has a limited restricted rotation, and therefore having it on the opposite side actually counts as a separate isomer. You can't just rotate it back into place. So for the ring as well, you can't really rotate the ring to make it, like you can rotate the thing that's in the ring and put it back into place, because it is literally in a ring. It is, if you rotate the ring, the ring will turn inwards and then it wouldn't work anymore. Or like usually doesn't work unless you have a very big ring, like at least 8 carbons in the ring, then only you can really do the insides thing with the ring, like the transis and then only you can do stuff with the ring and not have it break, I guess. Yeah, that's the point. Okay, anyways, now let's move on to Cairo Centers. What is a Cairo Center? A Cairo Center is basically something that allows, like, I don't really know what's the correct way to say it, but the thing is, you have like the carbon at the center, right? And it has 4 bonds. So all of the 4 bonds have something different on them, or at least in a way it kind of allows it to be mirrored, and the mirror image is cannot be rotated back to the same thing again. So that's the idea. That's the idea of a Cairo center. So in that scenario, it's usually the easiest way to find a chiral center is to see if the carbon having 4 bonds, or any other thing that has 4 bonds, can be a nitrogen with an extra dative bond as well, and it has 4 different things attached to it. If there's 2 similar things, then it doesn't work anymore, and that particular molecule doesn't really have like symmetry in terms of that particular chiral center. So that in that sense, you can then do the mirror image of it by just writing like what I showed you last time, and then you would get a stereo isomer of the same molecule in a way. Yeah. So yeah, that's how you figure out what's a chiral molecule. You just find how many chiral centers there are, and then probably have to answer MC question. Oh, are there 7 or 8 chiral carbons or something? Or how many isomers are there? Yeah, that kind of MCQ question. Or you might have to draw it out draw the stereo chemical formula. Okay, so yeah, so it shouldn't have a plane of symmetry, because once it has a plane of symmetry, it doesn't work anymore. You can rotate it back. So just in case you're a little bit worried about it not being a chiral center, just check whether you can actually rotate the mirror image back into the same chiral center. If you cannot, then it should be okay. But if you can, then no, you can just, it's not a stereo isomer anymore. Okay, so right, so what do chiral centers do, or at least chiral molecules? They can rotate plain polarized light, kind of. Of course, this has to be optically active. So what this means that the sample has to have more of one one chiral isomer than the other one. Like one of the isomers is compared to the other one. If they are 50-50, then they both kind of cancel it out and it doesn't have the same effect, doesn't have any effect on the light. So the optically active chiral molecule or the sample containing mostly one kind of isomer will then rotate plane polarized light in a certain way, in a sense, yeah. And so each of them will rotate it in opposite directions. That's the idea. You don't need to know anything else apart from this when it comes to the light part. Yeah, just know that they rotate it in opposite directions. And yeah, that's why in a way they're actually the same thing except for the fact that they rotate the light in the opposite direction. And that is what you need to know for A levels. I know there's something here that says usage of the term diastereomers is not required. I actually don't really remember what a diastereomer is. Sorry. But I think this one is something to do with the E and Z nomenclature thing, so yeah. But anyway, you don't need to know this for A levels, so it's not an important thing. I should probably go read it up. Oops. Okay. So next one is recognize that enantiomers have identical chemical properties as well. So the first one is that they have identical physical properties, but they also have identical chemical properties, except in the way that they interact with another chiral molecule. Okay, what this means is probably related to the shape of the molecule and how they can interact with each other. So this could be something to do with, for example, steric hindrance or something like that, like the forward attack versus backward attack, and we will see that in one of the topics, I think. Yeah, there's one mechanism for that. Okay. Yeah, so that's just what you need to know. You just need to know that they have identical physical properties and chemical properties, except for the fact that they rotate light differently and they interact with other chiral molecules differently, slightly differently. Yeah. Okay, so different stereoisomers exhibit different biological properties. Okay, so this is the important thing. You know, just now I mentioned that they interact with the other chiral molecules differently, right? That means that it is likely that they could be interacting with certain proteins or whatever in our body differently, because that's how biochemistry works. You have like two molecules interacting with each other, then something happens. Yeah, that's like the most lame way of describing it, but that's pretty much what happens most of the time. So many proteins actually function by having stuff that is complementary in shape. It doesn't have to be an enzyme. It can be a receptor. It can be some other kind of protein as well. The stuff that's on the how do I say it? The signaling receptors, the ones that are part of the phospholipid bilayer in the cell. Yeah, cell membrane. Yes. Okay. Yeah. So because of all these proteins, different drugs may react differently with the proteins. And in this scenario, that one of the scenarios that they very much talk about in lesson is there's this, I don't remember what it is exactly, what it's called, but one of the enantiomers is dangerous and one of the enantiomers is okay to use. So it was something to do with morning sickness. Yeah, it was that thing that's used to treat morning sickness, and basically they were selling it, but the thing that they were selling had quite a good amount of dangerous stuff, and it caused a lot of trouble, and it made the kids have defects. So they realized that late, and then had to make sure only the good one is produced, not the one that causes trouble. So do recognize that they can have different biological properties. So let's, like in a normal chemical reaction, if let's say there's no chirality in the other molecule, it doesn't make any difference. But if in a biology sense, there's definitely going to be other chiral molecules in the body, or at least in terms of protein, they are very particular about shape. So the stereoisomerism is quite important when it comes to how the molecule interacts with the protein. I don't know why I'm saying this with such significance, but okay. So Next one is deduce the possible isomers for organic molecule of known molecular formula. Okay, so yeah, this is what you do after knowing all the stuff that we just mentioned, like what kind of isomers exist, and yeah, like what are the characteristics of finding each of them. So how would you approach a question that asks you to find a number of isomers, or maybe draw out all the isomers or whatever it is? Well, you just need to look at the molecule first of all. If it's not drawn out for you or whatever, then you should probably do that yourself. Do the displayed formula or the skeletal formula at the very least, because you need to see if there's any double bonding or if there's any chiral carbons, and drawing out the skeletal formula at the very minimum will definitely help you with doing that. And from there you can easily find out where all the different isomers are. If it's structural, you just have to keep drawing out different positions of the functional group and making sure not to accidentally redraw the same molecule just from doing the branching thing. Make sure a branch is actually in the middle and not on the end, because there's no longer a branch. Yeah, you know what I mean. And for the cis trans, you look at double bond or rings. And then for enantiomers or Cairo Centers, yeah, just look for the Cairo Centers. See which carbons are sp3 carbons, and see if all of the four things are basically different things. Yeah, like the four bonds are all different. Then it will work. It is a chiral center. The reason why you have to see for sp3 specifically is because sp3 is the one with four different bonds. If it's sp2 or sp, then it has less than four bonds, and therefore it's no longer chiral. In a way, it doesn't display that chiral behavior anymore. Okay, so the final learning outcome is identifying chiral centers and of cis trans isomerism in a molecule of given structural formula. Oh, okay, yeah, it's the same thing. Just if you have the structural formula, you can just draw it out, because usually you write it and then you have to translate it into the skeletal or displayed formula. Then from there you can find out all the chiral and cis trans. Yeah, it shouldn't be that hard. Okay, yay, we're done. Yeah, this is actually pretty. This is just another topic to help you navigate the rest of the topics. Yeah, so next one will be hydrocarbons, and that'll be next week. I'm so sorry. Yeah, but okay, if you found this video useful, please press the like button and subscribe. And with that, I would like to say thank you and bye.